Dr. Melanie Martin, although she again doesn't need my introduction. Many of you know her. She graduated with a BSc honors in physics from U of M, from this university, and then went to get 2 masters and 1 PhD in biomedical engineering from Yale University. She was a postdoctoral and associate scientist in biology at Caltech in Pasadena, California, before she came to Winnipeg in 2004. She is now an associate professor of physics at the University of Winnipeg and director of magnetic resonance microscopy center in radiology at the U of M. And therefore she is also an adjunct in pharmacology department. I think you have full appointment, but you wrote here adjunct.
It's adjunct.
Okay. Ben was saying, anyway, an adjunct in pharmacology and physics as well. She's also a core member of BME program, and her specialty is in the development of non-invasive magnetic resonance imaging techniques to diagnose and understand CNS system. Okay, please join me to welcome Dr. Martin.
So, an interesting bit of trivia based on the emails from the biomedical engineering list. I found out that, well, I was the first BME graduate from Yale, and I just saw from the email that we have our first BME graduate going across the stage in October, so it's all great going. Okay, so what I'm going to go through today is why use MRI, and I'll go through a bit of how MRI works. And then, strangely enough, I'm giving a talk tomorrow morning as part of neuroscience rounds and Manitoba Neuroscience Network. So I thought I'd give them just kind of the hand-waving quick bits of everything that I do, and instead change for you guys, going through a new imaging method that we're working on, and one of my BME students and some of my physics students are working on this. So I thought I'd go into sort of details on that one, because that's really my exciting one. We're going to get down to less than 1 micron resolution. We're going to be able to see axons and so forth. So that's all using this new oscillating gradient spin echo sequence. And from that, as I said, we're going to show you how we can determine the size of cells and axons, show you some of our preliminary Monte Carlo data and some of our phantom data. And then I'll go through the relationship to the diseases in our mouse models and the conclusion. So, how many of you have heard of MRI before? Yay, that number is going up. It's almost everybody now. How many of you have had an MRI before? Was yours clinical or research?
All of the above.
Me too. I can tell you I like clinical magnets much better than research magnets. Research magnets, the bores are really tight and they're very dark. Clinical ones, they've got lights and it's big. So this is the 9.4 Tesla magnet that I commissioned in Caltech. Just to give you a size scale, before this gradient insert was put in, I tried to climb in the bore to see if I could fit. I got into about here, and then I realized if I kept going and get stuck, I'd probably get fired. So I don't know that I could fit in the bore, but I probably could have. So why do we use MRI? Well, that magnet that I just showed you cost us $2.5 million American dollars, and that was about 10 years ago when the Canadian dollar was worth virtually nothing compared to the American dollar. The signal to noise that we get is quite terrible, and just to give you an example, if you take sort of one click of your camera, you might get a picture like that, and yes, that's the best of my ability to draw. But if you take one picture with an MRI, you get something like that. And so the smiley face is in there somewhere, but it's so noisy it's very hard to see. So what you need to do is take picture after picture after picture and average them together before you can finally see something. So the experiments are pretty hard to perform. So for me, I'm working with mice. For you guys, I could ask you to get in the scanner, well, if you could fit and hold still. I still haven't figured out a way to ask my mice to hold still. So for them, we've got to anesthetize them. When you anesthetize a warm-blooded animal, they can't keep themselves warm. So we also need to keep a water heating pad in there. So we start off with the bore this big. We get down to a mouse coil about this big, or actually sometimes more like that. In there, because they're anesthetized, we've got to watch their respiration, we've got to watch their heart rate, we've got the water heating pad to keep them warm. So we've got got to have that all set up. It's really tiny. I've gone to cancer care multiple times to have me build me little tiny things, and they just hate me now because they can't build them so tiny and make it all work. And then once they're in there, we've got to be careful. So if I were to come in, say, with my glasses, and I just walk in the room, they're magnetic. If I'm not holding tight, they're going to go flying. And they're going to go flying right to the center of the magnet. And what's the center of the magnet? Your brain, if that's what they're imaging. So we got to make sure that everything in the room is very safe and tied down, or not metallic, or not being able to be pulled by magnets. And then once we get the images, what do they mean? There's a white blob in the image. Is that a tumor? Is that a stroke? Did the person swelling? Is it the water heating pad that was, over here showing up over here? And basically, I can't have someone say, okay, I've got this mouse model, tell me what's wrong with it. You really have to tell me what's wrong with it, and then I've got to find a way to find it. So if that really was the story, I wouldn't be here. So the reason we use MRI, despite all these hardships, is that we can see inside of the body without actually having to do anything to the body. It's all non-invasive. The mice I have to put to sleep, but you guys don't. I don't. I don't necessarily have to use contrast agents. It's all in there. And so the reason MRI was first used in the clinic was because of Dr. Mosley. So way back in 1990, he put a cat in the magnet, and this is actually a picture of a mouse. And it turns out that as the cat had the stroke, or the mouse, or the person has a stroke, we can see it instantly in the magnet. So in this white circle is mouse brain, and you'll notice that half of it is gray and half of it is white. This white side is the region that's affected by stroke. So way back when, before this happened, it would take hours and hours before they could diagnose a stroke. Now they can diagnose it as soon as they can get you in a magnet. They can start the treatments going faster. So the outcome from stroke has really improved since 1990 because of this. Very few human images in here, but here's one of them to show you a nice one. So it's a cross-section going through this way. So there's the person's brain, nice contrast with the nose and the lips and the tongue and so forth. So how does MRI work? So I like to show this picture. It's the highest field strength MRI scanner in Canada. That's for imaging, not for spectroscopy. And it's located at what used to be National Research Council Canada. Hopefully we'll find out what's happening to it soon. And this is it. It's 11.7 Tesla. So I like to show it not only to brag, but because it helps me in the next slide for the model. So if we model that magnet just as simply a bar magnet, and we put our patient into the magnet, we're not actually looking at the patient or the tissues or so forth. We're really just looking at the water inside the patient. So what's the chemical symbol for water? H2O. So it turns out we actually don't care about the oxygen. All we really care about is the hydrogen. We don't care about the hydrogen atom. All we really care about is the nucleus. So that's where the term nuclear comes in. So if we put a burst of energy into the magnet, the nuclei will sing. Okay, they don't, but we'll get into that in a bit. And it turns out that the stronger the magnetic field, the higher the frequency the note sung. And that's all governed by the Larmor equation. So they're not actually singing. What they're doing is they're processing, and they're processing a frequency that's dependent on this magnetic field. But for this model, to keep it simple, we'll do singing. So that's great. Now we've got this resonance. We put in our burst of energy. We've got the nucleus. We've got the magnetic. So where does the image come from? So we put our great drawing of the person into the magnet. We put in our burst of energy, and the nuclei in the brain start singing. Well, that's great, except there's a problem because the nuclei in the feet start singing. And it's not just the left foot, it's also the right foot. So we need a way to tell between these different nuclei so we can make our image and figure out where they are. So let's say I want to image this square. What we could do then is we could put on a magnetic field gradient, say from front to back. So the gradient is a linearly changing magnetic field. It's really a small change. So the field over here is a little bit lower, and the field over here is just a little bit higher. Well, now the nuclei up front will sing at the lower frequency, and the nuclei at the back will sing a higher frequency. So this slide alone is the subject of a 6-credit-hour graduate school course at Yale, but you guys only have a few minutes, so we're going to do it in a few minutes. So I'm going to have you guys pretend to be hydrogen nuclei, and I'm going to take an image of this room. So I'm going to put on a magnetic field gradient. It's going to be slightly lower magnetic field at the front of the room, slightly higher at the back. And I'm going to come by and play a note, and so those at the front of the room are going to get a lower note, those in the middle will get something in between. between and those at the back, since I don't like people sitting at the back. I'm going to give you a really high one. So here I come. I'm going to play it, and you guys come along. So we'll do the first 3 rows maybe. Hopefully you guys can hear it from over there too. Perfect. Do the next 2 rows. Great. Last 2 rows, how high do you really want me to go? Awesome. So when I do this talk with the old folks home, by the time I get to the front row is asking me again. They've forgotten their notes and they need me to play it again. So anybody else here forgot their note yet? Good. Okay, so now I'm going to have you all sing together. You ready? Awesome. So I can tell where you are in the room. Those of you with the lower notes are up the front, those of you with the higher notes are at the back, and those of you somewhere in between are in the middle. So that's great, front to back. Now we have a problem of left to right. So what comes here is we put on another magnetic field gradient, but this time we just put a little pulse. So the frequency changes slightly, say lower here and faster here. And so we're just putting it on for a brief bit of time. These guys will get a phase, and these guys will get a phase, and they'll be out of phase. And based on the phase, we can figure out where they are. So instead of having you guys stand up and spin around at hundreds of millions of times per second, we're going to switch it, and we're going to do the wave, because the farmers out there finally won a game, and everyone's happy. So I'm going to run across the room. The wave will start here on the advanced side, and as I run by you, that's when you get your wave. Ready? There we go. Okay. So I haven't yet learned to watch and run at the same time, but if I could tell where you were in the room. Those of you who stood up first are on that side, those of you who stood up again are on this side. But the good news is, now I'm going to have you do it again, and you're going to sing your note as you do your wave. Ready? Here we go. So now I don't need to tell where you are in the room. I don't need to watch, because I can tell those of you who sung earlier here, those of you who sung later here, those with the lower notes are up front, and those with the higher notes are at the back. So that basically gets you your image, or something like it. So there. That is 1 6-credit-hour course at Yale done in 5 minutes. So what you get out is this, and this is actually really real data. There are real people who can understand what that means and get information out of it. I'm not one of them. It's called K-space. So you got low frequency, high frequency, low phase and high phase, and usually what you're looking for is right in the middle. But what we do with this is we apply a Fourier transform, and we get our image space. So this is a mouse brain, thinking this way. So its nose is up here. We're either just below or just above the eyes, so it would be right there. And that's the brain. So that's how MRI works. If you want to see some of the little tricks, you can come tomorrow morning with what we're doing. But that's kind of the lower resolution stuff and the general stuff. What I wanted to get into now is our hot topic that's going to help us see these axons. So it turns out that water molecules move. Well, all molecules move. Unless you're at absolute zero, molecules are moving. So the molecules in this glass of water, if I can hold it still, are moving. And it's diffusion. It's just kind of random motion because of the thermal energy. They bump into each other, and it's kind of like the random drunk walk. Their net displacement is 0. The water's not moving up or down or sideways, unless there's a hole. But the mean squared displacement is non-zero, because there'll be a certain number that'll move a little bit right and a certain number that'll move left. And what Einstein found is that the mean squared displacement is proportional to time. So if we were to start our stopwatch here and let this guy move around and he ends up here, that's the end of our time. That distance there, overall for all of them, mean squared displacement is proportional to time. The proportionality constant is called the diffusion coefficient. And this diffusion coefficient, it depends on the medium that it's in, and its magnitude is sort of dependent on the ease of movement in the medium. So for water, it's pretty high. For something like molasses, it'll be a little bit lower. So for water at room temperature, it's 2 micrometers squared per millisecond. For water at body temperature, it's about 3 micrometers squared per millisecond. So remember these magnetic field gradients. We can use them for something else. So if this magnetic field is uniform, then all the spins will have the same frequency, which is good because then we can use it our way, get the image going, and make a beautiful image. But if we put on this magnetic field gradient, we know, as we just went through this demonstration, that the field that they'll feel is dependent on their position. So now their frequency is dependent on position, just like when we went from the front to the back of the room. So if the spins were to move, their frequencies are going to change with time. So if one of you were to get up and kind of move to the back and grab a drink, your frequency will change with time as I'm doing the display, or the model, the image, and we won't quite get a nice image. So this was something that they studied way back when as something that was bad for the image and going to cause problems. But that's when Dr. Mosley found those stroke images, and it turns out this can actually be really good. And it's much better than we originally thought. So what we use typically is something called a pulse gradient spin echo sequence. This is a model of it. So we apply a gradient pulse for a very short time. So again, some of the spins, say the back of the room, will speed up a little bit, some at the front will slow down a little bit. So you'll get, if I just turn it on and turn it off briefly, those at the back of the room will have sort of a big phase, those at the bottom, front of the room will have a lower phase. The net phase is going to be 0, but the mean squared phase depends on position, so the mean squared phase depends on time. So here's where the time comes in. So we apply the gradient, we wait an amount of time, we apply another gradient that's equal and opposite. So if nothing's moved, nothing changes, the phase line's back up, and we get back to our original signal, except for relaxation. If there's diffusion, that's when we'll get a mean squared phase that depends on time and the diffusion. So the actual sequence has us put in our burst of energy, then a little gradient pulse, then we actually reverse the phase, put in exactly the same gradient pulse, and we measure the signal over here to get our phase and our diffusion coefficient. So there's a bunch of regions and regimes depending on your time. So if we were to say have a water molecule diffusing inside a cell, we start a stopwatch and stop it, and in that time the distance it moves, or it can move, is much less than the radius of the cell. It's not going to feel the membranes, and so it's going to look like we're measuring free diffusion. If there is a boundary, but it's perfectly permeable, so if the water hits it, it'll just keep going straight through. Again, what it looks like to us is free diffusion. If there's a boundary that has some permeability, so it might bounce through, and the time is above the time it would take it to move across the cell, they call that hindered diffusion. And if, on the other hand, our time is so long that it's moved pretty much all the way across the cell a lot, and the membrane is either impermeable or only slightly permeable, what we have is restricted. So we're going from free to hindered to restricted. It depends on either the size or the time. So what we measured is no longer a free diffusion coefficient, we're now measuring an apparent diffusion coefficient. So if we have non-permeable boundaries or we're using long diffusion times, we'll get what's called this apparent diffusion coefficient. And this apparent diffusion coefficient is going to depend on time. So we'll go through a simple model first. We'll have equally sized square cells of length L on a lattice of A. We might have a diffusion coefficient for the water in the cell that's different than the diffusion coefficient outside the cell in the extracellular material. Could be slower inside here because there's stuff the water has to move around, like the nucleus, the cytoplasm, that's in the cells. It's been so long. All that stuff in the cell. The water's going to move around it. So some people will model this with a slightly lower diffusion coefficient. So when we do that, if we start our stopwatch and stop it almost right away, the water molecules have barely moved, and so we'll get a free diffusion coefficient. Sort of, it's a weighted mean of the intracellular and extracellular diffusion coefficients. If we start and stop our stopwatch a little bit longer, so the water molecules have a little bit more time to move, this is the smallest space between the cells. So those ones are going to start going from free to hindered to restricted. So the diffusion coefficient is going to get smaller. If we go a little bit longer, those in the cells will start getting restricted and hindered. So the diffusion coefficient will get even smaller. And if we go for really long times, everything is now restricted or hindered. And so we'll get, basically it goes to a constant that's pretty small. So the schematic looks something like this. You have your apparent diffusion coefficient as a function of your diffusion time. You start off at the top, if you remember, it depended on the volume fractions of diffusion coefficients. You go all the way down to the end, and it's proportional to something called tortuosity. What tortuosity is really measuring is the ease of something to travel from point to point. So if it's going to go from here to here, that was really easy. But now if I put a big block in there, to go from there to there, it's got to go all the way around. So that's a large tortuosity, because it can't go here, it's got to go there. So that is a simple model with one size cell. What happens if we now take our model and we change the cell sizes? So we've got a really big one, a really permeable one, this one's actually supposed to be a lot smaller than that one. What happens there? So if we were to pick one of our diffusion times, we might have different kinds of diffusion going on in each of them. These ones might be free, this one hindered, and this one restricted. So then what we get then is an apparent diffusion coefficient that's then depends on the fraction of water molecules that are free, the fraction that are hindered, and the fraction that are restricted. And then you get the diffusion coefficients for each one. And so that's a model that most people will use. And as you change this diffusion time, the fraction of water molecules in each of those types changes. And so we get actually a different ADC that will depend on time. So for long diffusion times, which is what we usually use, most of the water molecules will be in the restricted regime. If we can get down to the short time, most of them will be in the free regime. And somewhere in the middle, you get hindered. So we can actually make a model that looks like this, and the fractions and the diffusion coefficients will depend on the size of the things in the sample. And there's some really complicated models that will go through this, and you can actually get the distribution of sizes in the sample as long as you step through enough diffusion times. So basically, you run with enough of these, you've got enough measurements over time, and with that, you can have enough parameters to fit everything in the model. So what are we looking at in biological tissues? Well, free water diffuses about 5 microns in 5 milliseconds, or 20 microns in about 80 milliseconds. Cells are about 5 to 50 microns. Axons in mice are much less than 1 micrometer. So the standard MRI sequence that I told you about, this pulse gradient spin echo, it requires the diffusion time to be 10 to 100 milliseconds. So we've pretty much gone at least the size of the cell. So the standard MRI really can't view this transition from restricted to free diffusion. We need to watch it change in order to get the sizes. So standard MRI can't be used. So why can't standard MRI go to these shorter times? We have these very, very small displacements, so we're going to get very, very small phase differences. They're going to be very, very hard to detect. I already showed you MRI has very low signal to noise. Now we get this really small displacement, it's gone. Lose the noise. So what we could do is we could just average and average and average and average, and we'd still be here next weekend averaging and averaging. Not going to work so well. Another way to get bigger phases, if you imagine if I put a larger gradient on in this room, the steps between the notes that I played for you is going to get bigger. So really the front of the room will have a really, really low note, and the back of the room will have a really high note. So now the difference is big enough that maybe we'll detect it. The problem is these gradients, they're magnetic fields. We've got to turn them on and turn them off really rapidly, especially if you're trying to make the time really short, on off real quick. dB/dt, electric field. Electric field on the body, going to be bad. So from people who really take diffusion sequences and push them, I've heard of my friends complaining their skin starts twitching. Some of them actually felt pain. And there was a really wacky study done in the UK. They must have different ethics than we do. They actually put people in the magnet and they kept increasing the gradients. And, okay, tell me when it hurts. Tell me when it this. And so that's what they're really basing the limits of how fast these things can change. And it turns out from that UK study, it wasn't dB/dt that mattered, it was B/dB/dt. So the higher the field strength, and I use very high field strength, especially compared to clinical, the worse it is, and the slower you have to go. So for those of you taking clinical scans, don't worry, there's all sorts of things programmed into the scanner to protect you from anything happening. For my mice, sorry to use. So we don't really want to have these big gradients. We don't want to be averaging all weekend, so we need this other method. And that's where this oscillating gradient spin echo method comes in. So, projector is up there. I don't think I'm going to be able to reach it, so I'll have to do it by hand. So basically what we have here is we have this positive going gradient pulse, and then we have this negative going gradient pulse. So we dephase the spins and we rephase them. So if there's no diffusion, they'd all be in phase again. Then we dephase and rephase, and dephase and rephase. So right there, we kind of got the experiment to kind of average 3 times. And over here, we got an average 3 more. And I just put 3 up here because it's easy to draw. I'm trying to remember, Cheryl was using 20 last weekend. So 20 here and 20 more here. So she had it averaged 40 times. So now it's not that we do one experiment and wait, and another experiment and wait. Now we've got here 6 experiments really quickly done. So what we can do is use these multiple diffusion periods to increase our phase difference, so we increase the difference in signal, the loss, and so we can get more accurate measurements. There's a teeny tiny problem to this, which is actually a bit causing some difficulties. Each diffusion period is not independent of the other, so it would be nice if we could just average them separately, but each time you dephase and rephase and then dephase, it depends on what happened in the last one. So what we can't do is measure the ADC as a function of time. We actually have to measure it as a function of frequency. So we can't use everybody else's model that looks at how the ADC changes in time and then just plug it into ours. We now need this extra step where we have to change the ADC as a function of frequency to an ADC as a function of time and then use their model. Or actually what I'm trying to do is skip it right away and do ADC as a function of frequency straight into how things change. So what we're doing is we're measuring the ADC as a function of frequency, we're plugging it into here somehow, working on the models. We've got some pretty good ones so far. So others have worked out how this works as ADC is a function of time. Our goal is to change it to frequency. And so we're starting with simple geometries and then we're going to go to a mouse brain. So we want to know how the changes in the volume fractions of the ADCs and the types of things change to relate to the sizes of simple structures, which is what we're starting with. And we're using this OGSE sequence to measure this ADC at very, very short time. So we're going very high frequencies. So what we're going to do is we're going to try to infer sizes of these cells in our samples. And our first goal, once we've gone with our phantoms and so forth, is to move to the mouse brain and do axon diameters in mice. So here are some of our models. And you can tell now I've been stealing these from my students who are giving talks. So we have a Monte Carlo simulation. So basically we have, oh, it's going to come up. I'll show you the pictures of how it works. So the geometries, there's parallel planes. So these are basically giant cement blocks the water can't get through. The water is moving in between the two. And so the only distance that's restricted is this one. So we can push them closer and closer and closer together and see if we can tell how far apart they are. We did homogeneous spheres. That's our first model that we actually ran in a magnet. So we've got water moving in the spheres and water moving between the spheres. Trevor and Cheryl went crazy and they put cell nuclei in each of these cells to see if we can have complicated structures and actually see differences in the signal. And I'll show you that. We're getting there, but we don't have a formula for it yet. And then this is our model of our axons. So we've got these tubes that are like nerve axons. We've got water in them and around them. And we're trying to figure out if we can find the diameter of those tubes. So yes, they're moving. But here we have little red water molecules. And what he's showing is an actual picture from electron microscopy of nerves. So we've got these axons coming out. And around the axons are myelin. And these water molecules are diffusing in them. So we've got each of them placed all over the geometries and they're moving around. And we can get signal from each of them and average them up to see what we can get in MRI. Here he's showing you kind of a 3D model of the water molecules moving around in between the axons. And here he's showing some method for diffusion sequence. So the molecule starts there, he picks a random length and a random direction, and then the molecule moves. So with all of that, we can get some signals. And so we started with the parallel planes. What we have again is the apparent diffusion coefficient versus frequency. So remember, frequency is like 1 over time. So at very low frequencies, we're at very, very long times, we get short ADCs. Very high frequencies, that's very short times, so we get very high diffusion coefficients because we're approaching the free diffusion coefficient, which is around, I said, 2 point something for water. So these two planes are separated by 5 and 10 micrometers, and it seems like about 1 kilohertz is the best place in that graph to be able to tell them apart. This one is 2 microns, and this one is 1 micron. We haven't even hit free diffusion in this 1 micron all the way up to 10 kilohertz. We kind of get the mean, which is what I'm looking for the 2 microns at around 2.5, 3 kilohertz. So we're aiming with that. Trevor's analytical models are shown with the lines, and from that you can tell how big these are. We've gone to those spheres. So again, 10, 5, 2, and 1 microns. Again, the 10 and the 5 easily pulled apart around 1, 2, around 2 to 3, and the 1 we haven't gotten to free diffusion yet. And these are the cylinders, so 10, 5, 2, 1, and it's the same range. And then ones were done with cosine. Why are these ones also done with cosine? There should be sine waves as well. The sine waves, we don't actually have formulas yet to fit, and I'll explain why shortly. So then we go into the spheres that have the nuclei inside. We don't have a model for them yet. So what we did is we kept the nucleus size the same, but we changed the cell diameter. And with that, again, as we run, we can see these asymptotic values that we're getting to up here are different for the different values. So the hope is we can find some formula there. The signal can tell them apart. Hopefully we can find a formula to tell them apart. And then what we did is what we're looking at for diseases is, so we have these axon bundles and there's a bunch of fibers in there. What happens as things start to change and there's disease and the density of axons change. So what we did for this one is we kept the axon diameters the same, but we changed the packing fraction from 0.6 to 0.9. So 0.6789. And with that we can see that again, even with changes in packing fractions and not sizes, we can tell the difference. So we do think we're going to be able to do this. So how did it work with the real mRNA? So very first thing to do, put in something you know and make sure it works. So we know what we should expect for a tube of water. So in with a tube of water, without diffusion, with diffusion, so that's how you fit the signal. We get this beautiful straight line. Theoretically, based on our temperature, we should have 2.2 plus or minus 0.2. We got 2.14 plus or minus 0.01. So we had it all working. So then we went on to complicated structures. We took these polystyrene beads and we went and centrifuged them in water, so we have them very packed tightly at the bottom. So they're 3 microns in diameter. The polystyrene, there's no water in them. The water is just between them. So we have to change our model slightly because all we're looking at is the water in between the beads. And with that, we have the computer simulation of the 2 different pulse sequences. And it looks pretty flat, but it turns out the equation there fits, tells us a lot from there. And for the 3 micron beads, we actually get 18.6 in our very complicated model. Close, but no cigar. Getting there. The real data. So the estimated bead diameter for the real data, we only have a formula again for this one, it's 4.6 microns. Pretty close to 3. This error bar is absolutely horrendous. And if you look at this graph, it's going from -2 up to +5, and we're expecting a value of around 1.5 in there. These error bars are like more than 100% of the value. I'm pretty sure, given how flat this is, that we're overestimating our errors when we're calculating the signal. So we've got to go back and fix that. And we've got to do that within a week because we're off to a conference to present all of this. So I think we're going to be able to get that error bar down tighter. So what's the difference between these two? Again, I mentioned that we have to do this as a function of frequency instead of time. To switch it to time, we need to take the Fourier transform of the gradient waveform and see what we get. With a sine wave, I'm sure you guys know this with engineering, you get a plus and a minus. With a cos, it's 2 pluses. So with a cos, we get the straight answer. With the sine, we get the plus and the minus. And with the delay, we actually get a 0 as well. So it's actually much harder to figure out from here. Although much easier to run a sine wave because the sine starts at 0 and ends at 0. A cosine starts at 1 and ends at 1. So this is a slightly, actually I think in the simulation, this is a perfect cosine. In the gradient waveform, this is it's called an apodized cosine. So what are we going to do with this when we go to mouse brains? Well, diseases like multiple sclerosis damage these axons, and so we want to be able to see this damage. So we aim to watch this axon damage as it occurs. We have a Cooperzone mouse model of demyelination. So with that, they eat a toxic copper chelator called Cooperzone. With that, the oligodendrocytes die, so the myelin, the nerves demyelinate. And then if we stop feeding them the Cooperzone, it'll come back. So we hope to be able to watch this and watch this damage. In a recent study that we just published, it turns out that the small axons are the ones that are primarily affected. So we're hoping that with this, we can actually not have to go to EM to see that, but we can see that with the MRI. And so we aim to observe this in live mice. First we'll go with the dead ones, then we'll go with the live ones. So I hope what I've shown you today is that MRI can infer the sizes of the structures if we measure the water diffusion inside of it. And what we need to do is observe the transition from the restricted, the long times, to the free diffusion to find these times. And this oscillating gradient spin echo, all those different sequences are the ones that allow this short enough time. And we can do this in biological samples. So I think I still have plenty of time. So I thought what I'd do is, well, thank everybody.
heard of something called diffusion tensor imaging. That's different than this, right?
It's somewhat different. So this, we're just going to put it on in one direction for now, across the axons, so we'll be able to get the axon diameters. But
Okay.
if you want the diameters and the lengths and so forth, then you need the other directions. So what happens with diffusion tensor is you put it on in multiple directions, and then you can see which way everything's going.
Oh, okay.
And so if you're around tomorrow morning, I'm going to show you some of our applications of diffusion tensor. So, but long-term goal, say Vanessa stays on for a PhD for 50 years or so, we'll get her doing that too.
I think, Melanie, you should offer a course in BME on these topics.
It'll be a lot of fun.
It would be, yes.
Yeah.
I really enjoyed your talk.
So did someone let out that I give prizes for my talk, and that's why you guys are waiting on the questions? Should we run to the game and do the questions after? Okay, question.
Let me ask you one thing. You were showing basically the modeling for 2D imaging.
Mhm.
Is there any perspective for MRI in 3D?
Yes, so that again gets with the diffusion tensor. So actually what I'm trying to do is bring you one needed first, just to pick it up with the one. So the 3D, so mostly what it's used for in diffusion tensor imaging is along the axon it's pretty free, so you get those high diffusions. Across the axon it's pretty small because it's restricted by those. So there's a big difference between along and across. And from that they can tell, if you put on with enough directions, you can work out which way the axons go. So they can actually do tractography. We've used it, the stroke is based on diffusion sort of in the 3 directions. We've used it for all sorts of demyelinating problems. We aim to move this to 3D because we're never going to be able to be perfect on every axon if we just do it in one direction, but one step at a time.
Okay.
No? Okay.
Maybe another one. As soon as you are doing this in frequency domain, what is your way to determine basically frequency range? Because as soon as you have to go from frequency domain to space domain, like the resolution you will get in space domain depends on the frequency one.
Yeah.
What is the criteria for that in MRI thing?
So again, part of the frequency, especially for the human scanners, is the faster that dBdt happens, well, it turns out B dBdt, the worse it could be for the patient. But for us, again, the shorter diffusion, smaller phase, so you can only go up so high in the gradients again, and you've got this. So what you end up running out of time is the relaxation time. So you have to kick the spins out of equilibrium, that's that burst of energy we put in to make them spin. They want to go back. So we have that amount of time to make a measurement. So we fully put as many time waves as we can in that time, and then we're done. So that kind of limits how long we could go, and then so the frequency, the faster we go, we can't put enough in there, we can't get enough decay, so we can't make our signal measurement.
I have a question.
Yeah.
MRI is considered as a safe technique, right?
Yeah.
On the mice, has anybody ever investigated what is the long-term effect of being exposed into the MRI scanner?
I'm sure lots of people have seen sort of the long-term, look for long-term effects. When I was in California, we were doing a developmental biology lab, so I had all sorts of embryos kind of developing in the magnet. So long-term in that their lifetime, they were in and out of the magnet a lot and nothing happened, but it was kind of short-term because the lifetimes of the mice, it's 2 years, the frog embryos, I forget how long we kept them. But we've seen no damage with them. The damage more from the MRI comes from the people who forget what they're doing and bring something in the room and bam, or the guy cleaning the floor and bam. That's kind of things. Or people not being careful with the gradients with the scanner as well.
No, I meant basically any effect,
Yep.
any stimulation,
Nothing. Nothing's
any brain
seen.
cells, neurons.
Have you seen anything on long-term
No,
effects?
never heard of anything.
Because I heard it from a neuroscientist colleague that the stronger MRI machines, like 9.5 Tesla,
Yeah.
he would have been cautioned to put your brain there.
So way
He
back,
was conducting a research, but he was saying that he wouldn't put his own brain in that machine.
way back when they were just starting to get the 7 Tesla human magnets, they now have 11.7 Tesla human magnet. But with the 7 Tesla human magnet, the wavelength is about the size of the heads. So they were worried they were going to just get a dark spot in the middle of the residence that was going to cause problems. No problem at all. And so again, it's with that DVDT. The faster you get into the magnet as they're loading you in, the dizzier you get. Faster you go in, the higher the field strength, it makes it worse. So they've got to carefully move the person in and out of the magnet, but other than that, it's okay. I haven't seen anything. Okay.
Mind you, we are giving direct electromagnetic pulses to the brain. So,
And you have long-term good effects.
well, in short term we see something, in long term we see the effect will disappear, but we don't know.
Okay.
Okay? Thank you again. Feel free to help yourself with sandwiches and stay around, socialize, and ask questions if you were shy of asking questions, or just socialize with each other. Okay? Exxon has a question?
Oh, what happened to you yesterday?
Oh, yeah.
You want to
Oh.
stop by? So it turns out that all these sequences don't just work the moment we start, unfortunately. So we have to get things working on some kind of biological system that doesn't make me upset, doesn't breathe, doesn't need to be anesthetized, does what I want it to. So fruits and vegetables. And my advisor made a mistake in my PhD program going, this stuff is not working on rats. You have to get it working on a biological system before you can graduate. So I caught him, and ta-da! My thesis is about fruits and vegetables. There is a chapter on rats, but the answer is a bit more. So, what I'm going to do for the game is I'm going to show you an image of a fruit or a vegetable. You're going to shout out what you think it is. The first person to correctly get it gets a prize. I need your help sometimes.
What is the prize?
Hershey's Kisses. So I need help sometimes figuring out who's the first. I can't always hear it. I think I have enough that there's a tie that we don't need to stick to the rules of one. For those of you who have played this game before, you're not allowed to play again. So some helpful hints. The images that you'll see are axial images. So for those not familiar with images, if you get a green bean, we're not going to see it in this direction. We're going to see it like someone talked about. We're going to see it in that direction. Sometimes the fruit or vegetable would be surrounded by water. These images were not taken for the game, strangely enough, well, most of them. They were actually taken for research, so we needed water. I've noted in most of them, I'll try to tell you in some of them. And then I have an orange box surrounding each image, and that orange box is 4 centimeters wide, so 4 centimeters by 4 centimeters. You're ready for your first one?
Celery.
That's a celery.
Good for you.
So the celery is the star of my PhD thesis.
Thank you.
It turns out, and of course I've become a vegetable expert as this goes on, these little dots around the edges are called vascular bundles, and those vascular bundles actually have the perfect properties that are what I was measuring in my PhD. These little ones, I don't know what they're called. They were completely invisible at 2 Tesla, so I couldn't see them, but now we can see them beautifully at 7 Tesla. So someday, maybe when Vanessa gives her talk, she'll be telling you all about what these ones on the outside are called. Okay, next one. This is kiwi fruit. Who said that?
Yeah, that is
Ladies. So I seem to have some lucky coin in my pocket. Every time I move to a new place, we get a new magnet, and we calibrate it with kiwi. Ready for the next one? That's water. Wait, I thought I heard it say louder. It's a carrot. All the way back. I can't throw. So it turns out the carrot is the other star of my thesis. This is called the core of the carrot, right here in the middle. And the core is the only part that had the special property. But it turns out it only worked for old carrots. New carrots picked directly out of my garden, not good enough. Next one. And actually, here's a little air bubble. You can see a little bit of susceptibility artifact. It doesn't look like a circle because the magnetic susceptibility is all different. It's a mushroom. Where did the mushroom come from? Thank you. So mushroom did not make my thesis, so these are real technical terms. That's the stem and that's the act. That's Say it again.
Yes.
It's a vegetable. It's green. Close.
Zucchini.
It's a zucchini.
Well,
That was you, right? And so the zucchini I like to show, this was jammed right into the coil, so it's right up on the outside of the coil. When you get too close, you can actually see the struts. Putting in too much signal there, it kind of leaves these little black spots around the edge. Next one. This is a tube of water around the edge. Louder. Smaller. Smaller. Line. Who said line? Okay.
line. Yeah.
So I'm trying to shove the line in the magnet. This one really was used for the game only. And it wouldn't fit.
Or it'd be the water
So I
source.
tried to dig through the lab to find a knife to cut it. And the surgeon got really mad at me, took it, got out a scalpel blade, and was very carefully cutting this line. And I was like, come on, I just need the line. So I took the line, and of course, still cut would not fit in the tube. So I just went and squeezed it in there. The surgeon left the room going, crying. You can see each one of the little pieces of the line, and this one, like, because I squished it in, it gushed over the edge. Poor surgeon. I thought I heard him back.
Banana?
It's a banana.
Oh,
Who said that?
I said, but it's my second time.
Well, you can still have another one, just to a friend if you don't want it. This is a hot
It's
one.
the peel, right? yeah.
Okay, so, having given this talk around for a while, turns out the fruit of the banana
So,
is divided into 3 pieces. And that's been true for every banana since the first time someone told me that. The peel of the banana, so these are little strings, has 5 sides. True for every banana I've eaten except those from California, and I still don't know why. Next one.
Cucumber.
bean.
Okay, start again because I'm pretty sure I heard it all in there.
Apple.
Smaller.
Canadian?
Plum.
Potato.
Apple.
Orange.
It's smaller than a peach. Smaller. It's an apricot.
Oh. What's a slip?
Just a slice.
Yeah, I couldn't get the whole thing to fit, so I had to take the pit out, and then I poured it. And if you're going to come tomorrow, you'll see the game again, please don't guess. But there's something called a wraparound effect. So this is at 0 phase, and that's at 2 pi phase. You'll see there's a piece that I missed from the bottom. Shows up again on top. Okay, we're getting harder now.
Broccoli?
Smaller than lettuce.
Cabbage.
Smaller.
Cauliflower.
Smaller. 4 centimeters.
Broccoli.
Smaller.
Brussels sprout?
It's a Brussels sprout. So I tried to do an axial section of Brussels sprout, but there just wasn't enough water in these leaves to get anything. Okay, getting harder. That's water, and the outline of the vegetable is right there.
Diamond. Diamond.
Small though, remember, that's 4 centimeters. Lychee? Lychee? Lychee?
Green,
Michi.
it's a vegetable.
Potato.
It's not part of the vegetable you're used to looking at to identify what it is. It's a broccoli stem. Who said that? Okay. Close. So the broccoli stem almost made it into my thesis. This little white ring at 9.4 Tesla is very beautiful and very wide, and it probably would have made it into my thesis if I was using a 9.4. But I was using a 2, and that white ring just was 1 pixel wide, so it didn't quite make it. A couple more. Nice and loud.
Apple.
Turns out for apples, the air around the seeds causes such a bad susceptibility artifact that they don't make good images.
Didn't care.
What came from over there? Nope. So the Americans made a lot of make this a vegetable, but I think it's actually fruit. It's tomato.
Oh.
It's one of those Roman tomatoes. Okay. And so here
I
comes
would
four
think that they would
technical
be more
terms.
like
This
an
is the
engine.
fleshy part.
I guess
That's
it's all
the gushy
very
part,
good.
and those are the seeds. Okay, last one, and I analyzed personalities for free with this one.
What's that?
Watermelon.
Small, 4 centimeters.
Strawberry, probably.
It's a broccoli. So many people had seen the stem and want to know what the top was. Is that you again?
That's pretty.
Okay, so that's the last one. Thank you.
Yeah, the little bits.